So we'll start with the salivary glands. I think we took there, but anyways, just quickly repeat this. So we have three pairs of the salivary glands, isn't it? And the one is the parotid, which is the largest one, found anterior to the ears, right over here. Okay. Then we have the submandibular, right over here. Then we have the sublingual, just under the tongue, right over here. Right. This is the place where we have these three salivary glands. What is their function? They produce the saliva, right? And this saliva is very, very rich in what? Is rich in, do we have lysozyme in it? Yes. Do we have immunoglobulins in it? Yes, IgA. Antibodies, yes, of course we do. And most importantly, we have what? The salivary amylase, which can digest what? It can digest carbohydrates. So can we say that the carbohydrate digestion starts from the mouth? Can we say that? Of course we can, isn't it? So partial digestion of carbohydrates, your complex carbohydrates which you eat, so they're converted into like disaccharides or oligosaccharides in the mouth. And from oligosaccharides, when they reach to the stomach, oligo will be converted into disaccharides. Okay, like the sucrose. Okay, and then finally, or fructose. And from fructose, they will be converted into what? The monosaccharides, like the glucose, maltose, right? First, so it's basically, it's like a process, okay, which we'll talk about once again. Okay, so first, there are complex carbohydrates, then oligosaccharides, then you have disaccharides, and then finally glucose, like the monosaccharide. Right? That's what we want at the end. Okay, and what is the function of salivary? Other than that, it keeps our oral cavity moist. Also rich in what? A lot of mucus and the serous fluid. So it also contains the mucin because it has mucus in it, so it contains the mucin. Okay, and very important, when we eat the food, when we chew the food, right? So that food has to be binded together. So with the help of tongue, the food is mixed with the saliva, and it's moistened together, formed something called a bolus. What is a bolus? Bolus is like a semi-digested food, which is chewed or masticated. What is mastication? Mastication is chewing of the food, right? And then when saliva mixes with it, it forms it's formed a semi-solid mixture, right? Which is called as a bolus, which with the help of tongue we swallow down. Swallowing is called as deglutition, is a word for the swallowing. So all these words you should know. What is a mastication? Chewing of the food. Deglutition, swallowing the food. Is it? Buccal cavity is the mouth, right? This oral cavity. And then function of pancreas. Pancreas, see, we are doing right now the accessory organs. We know that not just having mouth, pharynx, food pipe, stomach is not enough. Or small intestine, large intestines. We also need the help of we're doing accessory organs, right? So we discussed about teeth, then the glands, salivary glands, and now the pancreas. Then we'll discuss gallbladder. Okay? So all these are called as accessory organs. That means without them, they do not belong to digestive system. But without them, digestion is not possible. And pancreas is so very important, right? Because pancreas, if you remember, we spoke, this is the pancreas, right? It contains two types of cells, SNR tissue and the alpha and the beta cells. Alpha beta cells producing what? Glucagon and the insulin. We're not interested in that. We are interested in the digestive function of the pancreas, which is what the SNR tissue is producing. The SNR tissue has an exocrine function. It will produce the juice through the what? Look at this, through the duct. Through this duct, the juice is released where? Here we have a stomach, So food will come from the stomach here, right? So the heavy food. So digestive juice will be released over here, the pancreatic juice. So what will your pancreas produce? It will produce two types of juices. The pancreatic juice, which is rich in bicarbonate ions, alkaline-rich fluid. Why do we need alkaline-rich fluid? Because the contents which are coming from stomach, they're all what? Acidic. Isn't it? Yes. So the contents, when they reach over here, if they're still acidic, halas, then no enzyme can work on them. Because the pancreas produces the enzymes. If you remember, we spoke about how important pH is for the enzyme. Then we spoke about in biochemistry that any higher pH or lower pH can destroy and denature the enzyme. So how can you expect the pancreatic juice, which will produce what? It will produce the enzyme-rich fluid, which has lipase, amylase, protease, right? So many trypsin, chymotrypsin, peptidases. And how can you expect these enzymes can work in the acidic environment? So it's very important for the pancreas to release a juice, okay, which is a alkaline rich, which is a lot of bicarbonate ions. So imagine if you have a lot of bicarbonate ions, right? Obviously the pH will go what? The pH will go up, isn't it? There will be no more acidic pH. Second point, the same pancreas will also produce a juice called as a pancreatic juice, which is an enzyme-rich juice. It will have all the enzymes which can digest all types of food, whether we eat proteins, whether we eat carbohydrates, whether we eat lipids. So very important point, everything is digested here in the small intestines. Why? Just because your pancreas produces all kinds of lipases, amylases, proteases, which can digest all kinds of food, whether it's a protein, carbohydrate, or the fats. That is an importance of the pancreas, isn't it? And then we also spoke about, if you remember last semester, we spoke about the two hormones. Does anyone remember the two hormones? Cholecystokinin and the secretin. I will show you the table. Maybe I have a table here. Remember this table? This was in chapter number 9. But there the color was blue. The table is the same table from chapter number 9. Secretin. It stimulates the pancreas to produce what? Bicarbonate-rich juice. Cholecystokinin, what does it do? Stimulates pancreas to produce the juice which is rich in enzyme. It will also stimulate, what is duodenal papilla? We'll go back to the diagram. Okay. Do you see this cup-shaped organ here? This one? This is called as a hepatopancreatic ampulla. It's like, this is like this. It's like a big cup-shaped organ. A duct, you can say. Okay? So what happens? When pancreas will produce the juice, it comes and it's stored over here. And when gallbladder will also release the juice, it will also come and it will release here.
Doctor, what's the function of the juice for the pancreas?
I said digestion of proteins, digestion of carbohydrate, digestion of lipids, kullu gets digested here. Everything. Not only for one thing. As I said that the digestion of everything completes here in the small intestines. We have these enzymes, and then we'll also talk about the brush border enzymes in a while. So finally, khalas. Everything is digested and absorbed right in the small intestines. That's why small intestines are very, very important. Right? This is the first part of the small intestines, duodenum, jejunum, and ileum. So duodenum is the most active part, where all the digestion would take place. Then as the food moves, then it gets slowly absorbed. We spoke about villi, remember? And the microvilli. How it's absorbed there and sent to the blood. You understand? So pancreas are very, very important. Okay, and as I said that this, what is hepatopancreatic ampulla? It's a duct where you store what temporarily, okay? You store the pancreatic juice or bicarbonate juice or the bile. Bile we'll talk about what's the function of bile in a while. Okay, and then we have a papilla, a small hole over here. Once the food will come from the stomach over here, right? Because of the action of the secretin, the hormone, and CCK, we just said CCK relaxes these sphincters. What are the sphincters? Sphincters are like the muscles which contract and relax. So when there's no food, the muscles are what? Contracted, not allowing anything to go in the ampulla. Khalas. But when the food is here, the duodenal cells, these cells over here, they produce CCK and the secretin, the hormones, which will cause these muscles to relax. Juice can come out. The papilla to relax, juice can come out. So it's a combination of everything. It's a combination of hormones like gastrin, secretin, cholecystokinin, hormones. Number 2, the pancreatic juice and the enzyme-rich juice of pancreas, right? And third thing which we'll talk about, the brush border enzymes in a while. And now the liver. The liver is also an accessory organ, isn't it? Right? Liver is also an accessory organ, largest gland of the body with more than 300 functions. We are not worried about other functions. We are worried about here what? The digestive functions of the liver. We know that liver produces bile. This is something which is very, very important. Right? Bile is produced. Filtration will take place here. Purification will take place here, right? Modification of food will take place here. Storage of food will take place here. Remember? Glucogenesis. Okay? So, this digestive function of liver is very, very important because it produces what? It produces the bile. Okay? Here's a diagram. Okay? Because it produces the bile. Bile is produced by the liver. Okay? This is a diagram from your book. Then through this, right and left, the hepatic ducts, bile is taken where? To the gallbladder. Okay? When CCK is released, cholecystokinin, remember the hormone CCK? CCK causes what? Contraction of the contraction of the gallbladder. Gallbladder is contracted, squeezed, and the bile is now released. Remember these ducts, all of these ducts. This is a bile duct over here, right? This is a common bile duct. So the common bile duct, bile also reaches where? To the ampulla, hepatopancreatic ampulla, and through the duodenal papilla, it is also released in the small intestines, in the duodenum. What does it do in the duodenum? What is the function of bile? Yes, online ladies? Anybody? What is the function of bile? What does bile do? What is the composition of bile? This mouse is giving me a very hard time. I can't even look at the... Digestion? Digestion of what? Haifa, Noor. Break down fats? Does it break down the fats, Noor? No. It doesn't only break down what is the important job done by the bile. I think we did that in biochemistry as well. What's the composition of bile? Bile contains, most importantly, bile salt. This was a question also in the midterm 1. What is the function of bile salt? Bile salt acts as the detergent, isn't it? This was a question biochemistry in midterm 1. Bile contains bile salts, especially it contains phospholipids also, but mostly bile salts. Okay, it contains bile pigments, bilirubin, cholesterol, but we're not interested in all that. We're interested where? In bile salts, which we get from the cholesterol. So what are these biles? So what do you mean when they act as detergents? They act as detergents. That means they are able to digest the fats. I need a plain. Do they digest the fats? Yes or no? Yes, ladies? Yes, Dada? I was having a plain page here. Does, yes, tell me, anybody? Yes? I'm just trying to open a screen, a plain screen. Nobody? White screen. Let me see. Break down the fats. Bile causes emulsification of fats. What is emulsification? It, for example, if I have, you know what's a triglyceride now, right? The moment when I eat fatty food, it's rich in triglyceride, biochemistry, right? Fat. It reaches to my small intestines. My duodenum, small intestines has an alkaline environment. I mean, it's all watery environment. Do you think they can survive in the watery environment? No ways. They need to be emulsified. So we will have bile produced, which will contain the bile salts. I can't use another color over here. There's some problem with this board. So let me try if I can use from here. Oh. And our screen is gone. Anyways. Okay. I'm just trying to use another color. They can use. I want to use this pen. It's not allowing me to use this pen. Anyways, I cannot. I cannot use this pen. Oh, where did it go? Anyway, let me start it all over again. Okay, let's say these are triglycerides. So I said they're emulsified. What does that mean? We have bile. Bile contains bile salts, right? So bile salt is something like that. Let's say this is a bile salt. It has two parts. It has a hydrophilic part. Okay. This is its. Remember phospholipids in the plasma membrane? Something like they have heads and they have tails. This is hydrophilic and this is hydrophobic, right? Something like that. This is the hydrophilic part and this is the hydro. What is hydrophilic and hydrophobic? Water hating and water loving. So this is what? This is a bile salt. Okay. So what will happen? This bile salt will cover each and every particle of what? Of the triglyceride. Can you tell me what is happening over here? What is happening here? The bile salt is covering what? What is it doing? It's covering the triglyceride, the fatty molecule, so that the hydrophobic is from the inner side, isn't it?
Yes.
And hydrophilic is from the outer side. So each fat particle is happy where? In the aqueous environment of what? Of the small intestines, isn't it? Making them what? Solubilizing them. So we can say that emulsification Can we say solubilization? Yes. Solubilization is also the job of what? Of the bile salt. Emulsifying the lipid, solubilizing, making them happy in the aqueous environment so that they become accessible by the enzymes, produced by the pancreas. Right? So every triglyceride, every fat molecule will be coated by the bile salt. That is why we say that it acts as a detergent. The way it is something like, I don't want to go into details for the mycel formation. The way when you put your clothes, in water, you add detergent. The detergent acts on the clothes and it removes the dirt from it. There's a mycel formation there. Similarly, we have a mycel formation here, right? And now what will happen? So does that mean the bile salt or bile have digested the fat here? They're still not digested. They're still triglyceride. They have not converted into what? Fatty acids or monoglycerides. Not yet. Then you have pancreas over here. Your pancreas over here. And pancreas will produce what? Pancreatic lipase. So this lipase will now what? Yes, it's going to break it down. Into what? Into smaller what? Fatty acids and monoglycerides. Into monoglycerides and fatty acids. Can you say now the bile causes digestion of the fat?
No. It's only making
Emulsifying and solubilizing. Or maybe breakdown of bigger particles into smaller ones. But khalas, not digesting, not converting triglyceride into monoglyceride, no ways. Not converting triglyceride into simpler fatty acids, no way. That's the job of the lipase. Lipase is doing that, not the bile. This is a common mistake done by students. Okay? Is it clear?
Yes.
It helps in the fat digestion. The right word is it helps in fat digestion. Okay? This is the real procedure. Then after that, when we have one more process, I don't want to go into detail. Then if you remember, we studied chylomicrons in biochemistry, which transport the fatty substances through the blood. Remember chylomicrons? Right? So these monoglycerides, if you see, then we have intestinal. This is intestinal. I don't want to go into these details. These monoglycerides will be again combined to form triglycerides. They will be coated by what? They will form the chylomicrons here. The biochemistry, you don't need to know all that. So chylomicrons will then, we have the lacteals over here, or lacteals. I will show you the diagram. What are lacteals? Wait a minute. Online people, are we okay? Are you able to understand what I'm trying to say? How fats are digested? It's both important for biochemistry as well as here. We had a picture of lactate. Look at this one. Right? So then in biochemistry, we spoke about how are fats transmitted through the, remember HDL biochemistry? High density lipoproteins, low density lipoproteins. Similarly, we have chylomicrons. So they have, they kind of, inside they form a phospholipid coat. Inside you have triglycerides then, fatty molecules. And then these chylomicrons will enter where? The lacteal. What are lacteals? Lacteals are the specialized what? Specialized? What are green colored things? Lymphatic vessels.
Oh, yeah.
Remember I told you the membrane is far apart. They're very permeable. So chylomicrons are bigger in size. They cannot enter into this vein or into the artery. Because arteries are, cells are closer. But lymphatic vessels are far apart. So chylomicrons will enter here, right? Through the lymphatic system. Then remember the lymphatic system collects all the lymph and brings it back to the subclavian vein. So then your chylomicrons will be delivered to subclavian vein, and that's how the fats are sent to the blood. So they are not absorbed directly in the blood. They're absorbed through the lacteals, the specialized lymphatic vessels. Is it clear? You understood the fact? As we eat, then bile is released. Bile contains bile salt. Bile salt acts on the triglycerides. Triglycerides are emulsified, solubilized. Pancreas produces pancreatic lipase. Triglycerides are converted into monoglycerides and fatty acids. They move into this. This is intestinal flora, right? The cells of the intestines. They are packed in the chylomicrons. As they're packed in the chylomicrons, from here they enter into what? The lacteals, the lymphatic vessels. From lymphatic vessels, they enter finally into the blood. That is how fats reach to our blood. Can they? Why do we need to pack them? Biochemistry question. Why do we need to pack them in chylomicrons? This can be asked in biochemistry. Why do we need to pack them in chylomicrons? Because fats are hydrophobic. Blood is not hydrophobic. How can your fats dissolve in the blood? They need a carrier. So chylomicron is a carrier. The way cholesterol is transported by HDL, LDL, like a sayara, taking them here and there. Same with your chylomicron, taking them here and there, like a sayara. Cool?
Yeah.
Online people, did you all understand? Digestion of fats. It was a combination of biochemistry and this as well. A revision for biochemistry as well as here. So here nobody will ask you about chylomicrons. I just wanted to collaborate the two things so that if the question comes there about chylomicrons, you know that chylomicron is there for the transportation of fats into the lymphatic vessels and then into the blood. Okay, all clear. Okay. That was about composition of bile, we did function. Okay. Same as the process for the fat soluble vitamins, of course. The fat soluble vitamins, they are not soluble in what? In the aqueous environment. So same process for them as well. Okay. Vitamin K, D, E, and A. Okay. And then we discuss this. Right. Then secretin, look at this. Secretin has a function on liver as well. The same chapter number 9. Remember I told you? It causes liver to produce more and more bile. And cholecystokinin does what? Contracts the gallbladder so that the bile is released. It also relaxes the dural papilla. Remember hole I showed you? The papilla hole through which the juices come out. It relaxes that so that the juice can easily come out. Now, have you ever heard some people have gallstones? Stones in the gallbladder?
Because they don't drink water.
Yes, that is about the kidney, but what causes the gallstones? They have gallstones. Gallstones are sometimes formed when they eat a lot of cholesterol, a lot of laham, right? There's a lot of cholesterol in the body. And some, because of a lot of cholesterol, so some bile salts get crystallized. You know what is crystallization? Chemistry, you know what is crystallization? They crystallize and form what? Small rocks, stones, called bile stones. And God forbid, if there's one stone over here, do you think the bile can come out? It has blocked the way. Bile cannot be released. And plus, CCK is the hormone which is continuously what? Contracting the bile. Isn't it? What will happen then? What's going to happen? Then these people will have lot of pain because CCK is contracting the gallbladder. But there's no place for the bile to come out because there's a stone. So they will face a lot of pain. So sometimes we give them medication to dissolve the salt, this stone. Sometimes you need to crush the stone with a laser treatment. Sometimes when there's no option, you need to remove the gallbladder. So if you remove the gallbladder, can they eat fatty food?
No.
For some time they cannot eat because
Salivary.
the problem is with the gallbladder, not with the liver. Liver will still produce the bile. It's not, see, gallbladder is not producing bile. It's just storing the bile. It's concentrating the bile. You understand? So liver is still producing the bile, but then what happens? Within few months, you will see that they are this one. This will become larger in size. No? It will become like this. So this will start storing what? Storing the bile. You understand? You know what I'm trying to say? So even though they might have a problem, doctor might tell them, do not eat fatty food for some months.
Yeah.
They might get diarrhea, loose motions because of fatty food. But after some months, this will assume the role. This will take the role. It will become like this, larger, and it will start storing the bile now. You understand? So they are all okay after that. So no need, because as long as liver is there, bile is still produced. We don't need to worry about that. Correct? Shall we move, ladies? Any question till now? Okay. Then this is, what are the 6 specific processes of the digestion? Let me see if anybody has any question. Okay. 6 specific processes of digestion. We have ingestion, means what? Placing the food in. So mouth is doing 2 things. Chemical digestion and physical digestion. What does that mean? Physical means chewing the food, grinding the food, mastication of food. And plus the saliva has the enzyme. So chemical and physical both. Right? What about pharynx? No digestion. Esophagus, no digestion. Stomach, again both. Chemical and physical both. Because chemical, like protease acts there. And physical, because you're churning, look at my hands. It twists and turns your stomach. So physically food is also break down in the stomach as well. Okay? So remember, in the mouth and stomach, we have both chemical as well as a physical digestion. Very important question. What is the difference between the peristalsis and the segmentation? I've seen most of the students, they are confused with it. What is peristalsis? Peristalsis takes place everywhere in the gut. What do you mean everywhere? Starting from what? Esophagus to the anus, everywhere. You understand? It takes place in the esophagus, in the stomach, in the small intestine, in the large intestine, and everywhere, kullu. And what is the function of peristalsis? Like alternate contraction of what? Remember the inner circular muscles and outer longitudinal muscles? Contract, relax, right? And food passes from here, look at this, from here to here now. So pushing the food, like I swallowed. From esophagus, food reach to stomach. How? Peristalsis. From stomach to small intestines. How? Peristalsis. Small intestine to how? Peristalsis. Like everywhere, kullu. But then segmentation is different. It's more common where? In small intestines. More common here. What is its function? It doesn't take place everywhere. More common in small intestines. Its function is not to push the food forward, go forward. No. Look at me. Segmentation is like I'm moving here and there. Did I move forward? I'm just moving at one place here and there. Back and forth like this. If I move like this is peristalsis. If I stay at one place and simply move here and there, mixing the food is the segmentation. Mixing the food with the digestive juices is the segmentation. As you can see here, this may be enzymes, this may be food, like mixing it here and there. And now everything is mixed together now, isn't it? So its function is not to push. This was a question maybe a couple of years before. We gave a question about peristalsis. No, the question was segmentation. It was true and false, I think. Does the segmentation push the food forward? No. It's there to mix the food, not to push the food forward. Remember, take care of this. Online ladies, any question? Are we okay? Did you all understand the difference? But very important. Don't make a mistake in this question. Okay, good. All clear. Erase this. Okay. Now, as I said, food breakdown is very, very important, isn't it? Now we will see how various enzymes will try to what? Digest the food. We'll see that now, okay? I want you to go straight to this diagram. Very important diagram again. All 3 slides, very important. You should know all the enzymes. You should know the location, where carbohydrate is digested, where protein is digested, where it is absorbed, what enzyme. You should know all these things, right? The first slide talks about the what? The carbohydrates, isn't it? As I said, that we eat carbohydrates. We have in the saliva, we have the pancreatic, we have salivary amylase, isn't it? Partial digestion. Even then, in stomach, do we have carbohydrate digestion in stomach? Mafi. No, no, and no. Mafi carbohydrate digestion in stomach. Only protein digestion in stomach. Okay? Then it reaches to the small intestines. Okay? There we have what? Pancreatic lipase. So finally, we have oligosaccharides and disaccharides. Can you absorb them? No. You still cannot absorb them. You still have to convert them into what? Into fructose, sucrose into fructose, and then finally into glucose or galactose. Maltose, sucrose, lactose, disaccharides. Right? So sucrose will be converted, which is a disaccharide, will be converted into what? Glucose and fructose by the action of enzyme called as the sucrase. Maltase. Maltase will convert the maltose into what? To glucose units by chemistry. Lactase. Lactase will convert the lactose into galactose and glucose. And this is done by lactase, maltase, sucrase are the brush border enzymes. Dextrinase, glucomylase, lactase, maltase, sucrase, they are brush border. Now you will think that what are brush border enzymes? She never told us about that. Brush border enzymes, if you remember, I told you about remember microvilli? Yeah. Here the plasma membrane, here, there will be sucrase present. Right? So they're all converted into monosaccharides. After monosaccharides, they will be converted into where? They will be taken by the small intestine, by the microvilli, and from the small intestines, they will go through the hepatic portal vein, cardiovascular system. See, every chapter is now important because you need to connect all the chapters. In chapter 10, we studied through the hepatic portal vein. The products will be sent where? To the liver for processing, for only processing, storage, filtration, killing of microorganisms, remove the drugs or anything which is poisonous, then it will be released into circulation. That is about what? Carbohydrate. What are proteins? When I eat proteins, nothing happens in the mouth. They're still proteins. Tertiary structural proteins or quaternary, nothing will happen there. But the moment my protein reaches where? To the stomach. Pepsinogen is converted into pepsin by the action of hydrochloric acid, HCl. And this peptin starts acting on my protein, starts digesting the peptide bonds. And now I have large polypeptides. Finally, but it's still not digested. In stomach, it's still not digested. Partial digestion of protein in the stomach, very important, not full digestion. Only partial digestion. Now protein will reach where? To the small intestines. Pancreas will produce a pancreatic juice, which contains the trypsin, chymotrypsin, carboxypeptidase, right? And all these will be converted into smaller polypeptide chains. And the peptides, like maybe 5 or 6 amino acids will be there, 1, 2. Remember biochemistry? We still cannot digest them. We can digest either just one amino acid, or we can digest what? Dipeptide, or we can digest this. We can digest either amino acids, tripeptides, or dipeptides. So then again we need the help of what? Brush border. Remember the enzymes. Amino peptidases, carboxypeptidases, and the dipeptidases, which converts them into amino acids and tripeptides. And these are absorbed through the intestinal wall. In the intestines, they're sent again to the liver, to the hepatic portal vein. Fats, I already told you, emulsified by the detergent action of boiled salt. We discussed that. Then the pancreatic lipase, and then we have what? Monoglycerides and lipases. Then they are packed into chylomicrons. Then they go into lacteals, right? And from lacteals into the blood. That's how the fats are digested and absorbed. Remember this duct? Thoracic duct. We discussed that in chapter number 12. Thoracic duct. Right, finally into the subclavian vein, and they become part of the blood after that. Absorption, we already spoke about the mucosal cells. And defecation, what is defecation? Defecation is throwing food out of the body. Look at this. This is the defecation, isn't it? When you throw food, undigested waste out of the anus, that's a defecation. Now, what are the various activities which takes place in our mouth? Swallowing is called as deglutition. I told you already, right? I already told you no digestive function in pharynx and esophagus. So remember this thing. Mouth, yes, carbohydrates are digested. Esophagus, mafi. Pharynx, no, they don't have any digestive enzyme. Saliva is only in the mouth. So they are. Now, what is the function of esophagus or the pharynx? Simply passageways to send food to the stomach. The way we spoke about a respiratory system. We have pharynx, then we have larynx, then we have trachea, primary bronchi, secondary bronchi, tertiary bronchi, isn't it? Then we have terminal bronchioles, and we have respiratory bronchioles, alveolar duct, alveoli. Oh, all mean so many things. And then the air reaches to the alveoli. So same way, this is a passageway. Pharynx and so is a passageway to send food to connect the food to the stomach. Right? And in the mouth we have two phases, buccal phase and the pharyngeal esophageal phase. What is that? When we chew the food, bolus, the food touches the palate. Reflux is activated. Okay, so swallowing reflux, deglutition. Remember I told you uvula. We have that, this uvula. That's something like this, which is hanging at the back. So uvula and soft palate will cover my what? Nasopharynx. So that the food doesn't go into the nose. Then I have the upper esophageal sphincter here, this sphincter. It will open. And this is the bolus, right? Chewed food mixed with the saliva. It goes to the esophagus. It goes to esophagus, sphincter is closed again. Finally, the food reaches where? Another sphincter called as what? The cardioesophageal sphincter. We spoke about it last time, right? And now the food is in the stomach, and this is closed again. If there's a vomiting, at that time, this one is open.
Yeah.
Then only things can come out. There's a lot of acid, or if there's any undigestion, extra food, all that will come out. Right? So do we have a class? Should we stop here? Do we have a time? You take or no? Khalas? You have a class. Okay, no problem. We'll stop over here. I think tomorrow we'll study on because we just have 3 or 4 slides left. Okay? And we are going to stop. I'll show you there. We just have few things because all these things you already took them. Like this is the repetition, you know. Again, the same hormones, secretin and all. So I'll show you. We're going to stop. This is the last slide. Online people, this is the last slide. This is the last slide. So this is the, I will send the email also, don't worry. Okay? We're going to stop over it. After this, we're not going to do anything.
Then
119. Okay? I will send the email also, don't worry. So tomorrow we'll start. Tomorrow we don't have a class, do we? No, we don't have a class tomorrow. Tuesday, we have a class. And you know that did you get an email from Dr. Karyapar? From 1st of Ramadan, 13, all the classes will be online.
yes.
From 13 to 21, right? So that means on Monday we have biochemistry online, then Tuesday the class will be online, so you don't have to come here, right? So we'll finish this on Tuesday and start because I already started urinary system with other classes.
Tuesday should be online.
From Tuesday onwards, they're kullu online. So Tuesday, Wednesday, Thursday, we'll take online classes, okay? And whatever we finish till Thursday, that will be included in the this Thursday. So that will be in the midterm and the final. From Sunday, we'll not take new material.
Okay.
Sunday, Monday, Tuesday, we'll do a revision week. If you have question, you can join. If you don't have question, you can sleep. It's happy Ramadan. You can take rest, right? Because I will be just revising the concept. Even if I have one student, I will be revising the concepts, right? I will not count your attendance. If you wish, you can join last week. If you don't wish, don't worry about that. That is a revision week. Okay. All the best and happy Ramadan to all of you. Okay. Yes, online ladies, do you have any question? Thank you. Thank you, everyone. I will send you the message also. Don't worry about the slide number and the page number as well.